Hi, my name is Shereen Yates, and I'm the founder and CEO of Nima, and we are delighted to present a summary of how we validated the Nima peanut sensor, both at Nima with internal testing and third-party lab validation. Today I will be joined by Dr. Aquanet Burt, who heads our R&D department, and our CTO, our Chief Technology Officer, Francisco Diaz-Lorenco. As an agenda today, we're going to be discussing an overview of what Nima is about and why we exist. And Francisco and Aqua will be introducing the peanut product, the methods of validation and testing, and also the results. First, our mission at Nima. We enable people to be their healthiest selves by giving them the power to know what's in their food. We do this by creating people-friendly products and sensing technologies that identify individual ingredients in food. We generate and share data that tell people whom and what to trust, and we champion and support people who care about what they eat. As a product overview, we have a number of different components that make up the Nima platform, starting with a one-time use disposable capsule where users put a sample of food in, the electronic sensor, and then an app that's connected to the sensor where you can actually record your test results, where you ate, what you ate, and see what other people are also sharing on that platform. So Nima leverages antibody-based chemistry, and it works like this. First, you take a pea-sized sample of food and put it in a one-time use capsule. Then you screw the lid shut to grind the food. And once you do that, it triggers the chemistry. The chemicals in the capsule mix with the food, and then you put that in the sensor, and there's more mixing going on. And then the sensor images the test strip and displays a result. It's a binary result, so if there is peanut found, a peanut icon will appear on the device. If no peanut is detected in the sample, then a smiley face appears. Nima is one additional data point and access to a community of thousands in addition to everything you're already doing. So it's important to note for Nima that the test results are only representative of the sample placed in the capsule and in the Nima platform, and not a guarantee that the entire dish or food sample will have the same results of the Nima tested sample. Use of this product should not replace education, medical advice, compliance with food labeling, and adherence to avoidance strategies. Our community is using Nima wherever they go. We see people using it at restaurants, at special events, when traveling, and also at friends' houses. We've seen Nima really empower people with this one additional data point, and when they let Nima take the first bite, there's a lot of peace of mind and better data to make more informed decisions about what you can eat. With that, I'm going to transition to Francisco, who can speak to the product validation.
Hello, I am Francisco Diaz-Lorenco, and I'm Nima's CTO. And next, we would like to present the validation data for our peanut test. The Nima team has leveraged years of experience not only developing the Nima gluten sensor, but also other diagnostic products, and we've used that experience to develop the Nima peanut sensor. Nima used a combination of internal testing and external independent laboratories to perform the product validation. We have ran thousands of tests on hundreds of foods to determine the product performance and fit for use. Testing included a mix of packaged foods, restaurant foods, and also prepared foods with known amounts of peanut. And next, I would like to introduce to you Dr. Aquanet Burt, head of our R&D team, to discuss the Nima internal validation data.
62 food matrices were prepared here at Nima to cover a range of different components and processing conditions. These included nut butters, bread, cookies, and French fries. In 55 of these foods, peanut in the form of a NIST traceable peanut butter standard was spiked into the food to a final concentration of 10 ppm. In some foods, this spiking was done before processing or preparation in order to simulate incurred samples. In the remainder, for foods such as ice cream and cake frosting, spiking was done after preparation. The peanut-free version of these foods served as the unspiked or negative control. In addition, 7 as-is foods were tested without spiking, Of these 7, 2 were known to contain peanut, and the remaining 5 were confirmed as peanut-free by standard ELISA testing. All foods were tested on the Nima device in replicates of 10 and on 2 different capsule lots. Each capsule lot contained a different immunoassay strip lot and a different buffer lot. They were prepared with the same manufacturing processes but using different sets of raw materials in order to assess lot-to-lot variance. For comparison, these same foods were tested on ELISA. Shown is the summary of results obtained for each lot as well as data for both lots combined. The first data row shows that at 10 ppm, our true positive rate or sensitivity ranged from 98.2 to 99.8% for a combined sensitivity of 99%. Accuracy and precision stats are listed in the last 2 table rows. The accuracy for both lots was 97.6% and the combined precision was 96.3%. These performance characteristics are based on a spiking level of 10 ppm. Foods with higher peanut concentrations, greater than 21 ppm by ELISA, had a 100% detection rate when tested with both lots. In addition, all foods testing as positive with the Nima device were verified as having peanut above the LOQ of the ELISA, while negative samples tested below the LOQ. Some foods in the nightshade family, for example tomato and eggplant, gave false positive results with at least one of the 2 lots tested. These foods have been excluded from this analysis, and we recommend not testing these foods with the peanut device. To assess cross-reactivity, we tested 30 peanut-free flours and spices. Combined specificity or true negative rate was 91.8%. This analysis again identified nightshade foods, for example paprika and cayenne, as assay interferents. Sesame and tamarind were determined to cross-react. All cross-reactants and interferents identified may give false positive results, and testing should be avoided. We also recommend diluting flours and spices in order to prevent clogging of foods in the capsule.
To supplement our validation testing, Nima also used 2 independent laboratories to perform additional testing. First, I would like to discuss the work BioFront Technologies carried out. The purpose of the study was to challenge Nima with a wide range of quality control and reference materials. These materials are usually used to assess the performance of ELISA test kits, so we wanted to understand how Nima compared. For this test, various samples were sourced from FAPAS, DLA, and LGC. Each sample was tested as 6 replicates by 2 operators using 3 Nimas each. The sample matrices included items such as cookies, flours, powders, chocolate, and other foods. And in an effort to further challenge the specificity of the Nima sensor, blanks were selected which contained higher amounts of legumes and other nuts, and none of these samples yielded a false positive result. As shown on the table, 29 different samples were tested. 11 of these samples were expected to contain less than 1 ppm of peanut, or well below the level of sensitivity of most assays. In this group of samples, Nima correctly reported a no peanut found in all but 1 of the samples. Another 3 samples contained peanut between 2 ppm and 10 ppm, still below the limit of detection of the Nima sensor. But even in these more challenging cases, Nima was able to detect peanut in 6 of the 18 tests performed. And in samples containing 10 ppm or more, Nima correctly detected peanut in 89 of the 90 tests. The single false negative result was reported in a 15 ppm chocolate matrix sample. And as a reminder, at this point Nima does not recommend the testing of solid chocolates with Nima. But in summary, in this evaluation, Nima had a 98.5 accuracy rate for negative samples and 98.9 accuracy rate for positive samples. And next, we will review the results from another evaluation. This next evaluation was performed by Microbac Laboratories in Oak Ridge, Tennessee. In this evaluation, 10 different foods were prepared containing peanut at different levels, ranging from 5 ppm to 100 ppm, including blanks. Each of these foods were also quantified via the R-BioFarm Fast Peanut ELISA test to verify the amount of peanut in each sample. And as before, 6 Nima replicates were done on each food using 6 different peanut sensors. For all samples at or above the target LOD of 10 ppm, the Nima sensor had a 0% false negative rate. At the 5 ppm level, all tests except for one resulted in a peanut fan result. Nima was able to detect peanut in 5 of the 6 cookie samples and 6 out of 6 for all the other sample matrices. The Nima sensor also reported 2 false positives, 1 out of 6 with brownie samples and 1 out of 6 with a soy sauce sample. Combined with the 1 negative at 5 ppm for the cookie matrix, the false report rate was 0.9%. Testing with chocolate initially resulted in no results, or the exclamation point on Nima. Microbac successfully conducted further tests to optimize the processing of chocolate with Nima, but at this point, Nima will not recommend testing of solid chocolate samples. In summary, the sensitivity of the Nima peanut sensor in this test was 99.6%, while the accuracy was 99.1% across all ppm levels tested. The false negative rate was 0.37%, while the false positive rate was 3.7%. And at the target LOD of 10 ppm, the sensor had 0 false negative results. And in summary, when considering the high performance in the extensive internal testing and the 2 external product validations, we feel that the Nima sensor will be a useful and valuable tool for the detection of peanut in foods. In summary, the study conducted by Microbac highlighted the sensitivity and accuracy of the peanut Nima test. At the target LOD of 10 ppm, the Nima sensor had a 100% sensitivity rate, while the specificity was 95.8%. And overall, the Nima peanut sensor had an overall accuracy of 99.2%. And if you have any questions or require further information, please visit our Nima website or contact our support team. We will be happy to assist you.